episode of the BioHub. Today, we're delighted to be joined by Devyn Smith, who is the CEO of Arbor Biotechnologies and a key figure in gene editing and regenerative medicine. With over 20 years of experience in biotech and a PhD in genetics from Harvard, Devyn leads Arbor's efforts to develop next-gen gene editing tools. He also serves as a board member and former chairman of the Alliance for Regenerative Medicine, helping to guide the industry's future. So join us today as Devyn gives a run through his journey, the recent advancements at Arbor, and his role as board member and former chairman of Alliance for Regenerative Medicine. So Devyn, thanks so much for coming on. How are you?
I'm good. Thanks for having me, Tom. It's a pleasure to be here.
Yeah, it's a pleasure to have you on as well. So look, let's jump into a little bit about you then. So can you talk us through your journey to get to where you are now, and what inspired you to pursue a career in the biotech industry?
Yeah, so I grew up in a small town in Utah, so I have probably a more non-traditional biotech background, and in a large family of 11 kids on a farm. And as a kid, I was always fascinated with, we had a lot of animals, and particularly chickens, and as we'd breed chickens, you'd get different traits coming out. And so it fostered this real interest in genetics such that, we used to, you've got chickens that have feathers, these big hats, or you have feathers on their feet, and you can begin to breed them and generate all sorts of interesting mixes. And that, I think, really sparked a real interest in genetics and biology. And so, going through high school, I was very interested in the sciences. Went to college, and again was very interested in thinking about the biology and how do things develop the way they do, and eventually led to my PhD at Harvard, looking at developmental biology. So why, how do you go from a very simple cell structure to these complicated three-dimensional beings with thousands of different cell types? And so understanding the processes and genetics involved was interesting. So I did that, and as I was finishing my PhD, I was sort of at that crossroads people reach, which is, do you want to go an academic route, or you want to do an industry route, or something different? And I felt that at the time, I really enjoyed thinking about science and planning experiments. I really wasn't particularly enjoying the work on the bench. And so I actually, after a PhD, went into strategy consulting because I wanted to learn the business side of science. So I did that for about 8 years, learned a tremendous amount about the business side, from generic drugs all the way to new startups to big pharma and everything in between. And that was a great experience to help understand and learn more about the industry. I then left and went to Pfizer and in their R&D organization. And it was a really interesting time to be there. Pfizer had just done their merger with Wyeth, and were trying to figure out what was the next thing, what was Pfizer going to be as this combined entity. So had the opportunity to help with that, and then spent a couple of years in the UK in the first Cambridge, focused in on some of the cell and gene therapy assets that Pfizer had. And that was a fantastic experience to really broaden my depth and experience in the cell and gene space. And did that for several years, came back to the US and decided I really wanted to double down in that space. So I left Pfizer and went to a company here in the second Cambridge, Mass, called Sigilon Therapeutics, which was a company looking at taking induced pluripotent stem cells, turning those into beta cells for type 1 diabetes. And so Sigilon was there about 4 years. We did a partnership with Eli Lilly on diabetes, and really worked hard to bring this novel technology into the clinic. And the company went public, and soon thereafter I actually left and came to Arbor. Arbor is a next-gen gene editing company, and as I've been in this space a long time, I've been a part of ARM, which is as you said, the cell and gene therapy industry group for since the beginning days, so almost 15 years now. And I've always felt that cell therapy has a place and gene therapy has a place, but there's a tremendous amount of space in between that no one was covering. And I feel like gene editing has the opportunity to sort of fill that void, where we can now actually fit and fundamentally impact the DNA to provide curative approaches in the right area of the gene. And so I think that is fundamentally a game changer. It also allows us to move cell therapy to in vivo. And so I think that gene editing really is starting to revolutionize the cell and gene therapy space, and will continue to. So that's sort of the journey. I think biotech all along. I think there's nothing better tthan being in a career where you actually can impact the health of humankind, right? Like that's something that is always rewarding. And you hear stories from patients, and you develop new drugs, and you see the impact. Like that's rewarding. That makes it worth all the pain and suffering of they have to go through for drug development.
Yeah, and it's a similar. I mean, most people that I speak to have a very similar perspective on why they got into the industry. It's all about the patients. So good. And can you share a little bit about the origin story behind Arbor Biotechnologies and the inspiration behind its founding?
Yeah, so Arbor was co-founded about 8 years ago by two pretty prominent scientists, Feng Zhang, who is a CRISPR pioneer from the Broad and MIT, and David Walt, who had co-founded Illumina, among other companies. He is at the Wyss Institute in Harvard. And the two got together at the time, recognizing that Cas9, which was the original CRISPR enzyme that folks like Intellia and Editas and CRISPR use, but they recognized there are shortcomings, that Cas9 is not going to be able to do everything that we need it to do. And so they got together, formed the company to really identify novel, differentiated approaches to gene editing that allow us to really edit across the entire genome, as well as being able to do all kinds of editing, so that we can effectively build a toolbox that allows us to treat any genetic disease, no matter what the disease is. And so that was the ethos behind Arbor. I think as we look at what's been accomplished over that time, we now have a really robust toolbox that allows us to really tackle any genetic disease out there as a company, which I think is really important. So I think that's kind of the founding of the company. And I joined 4 years ago to take the toolbox and be very focused on translating that into therapeutics.
Good. Awesome. And Arbor were recently announced that you guys entered the clinic, and then also secured 74 million in funding. Can you talk us through the lead candidate, the tech, and how the funds will be used?
Yeah, that's an important question for us. So the technology we have and the way we've focused our efforts, our current pipeline, we have 5 programs. Our lead program is in primary hyperoxaluria, which is a rare genetic disease. It's in the disease is in the liver, but it impacts the kidney. So children are diagnosed with kidney stones, which children do not get kidney stones, but in this case they do, because there's a mutation where their bodies are unable to, they have an enzyme that's dysfunctional, so they build up this oxalate, which causes kidney stones and destroys the kidney. So these kids will have to have kidney transplants in their late teens, early 20s. So that's primary hyperoxaluria. Our approach there is to go in and do a gene edit in the liver to effectively eliminate the production of oxalate and form a different metabolite called glycolate, which is water soluble and can easily be cleared. And so effectively eliminating the disease state, we hope, and effectively in a one and done approach. So treat patients once, fix it throughout the liver, and they can live a more normal life. So that's the goal. The program, the data look quite compelling in both mice and non-human primates, and we got clearance to start the study in the United States and in the UK, and the trial is underway at this point. So it's exciting to be able to take what will be the first non-Cas9 tested in vivo, as well as we believe the first CRISPR approach to actually treat pediatric patients. So a lot of firsts with this program that will be very exciting. So that's the lead program. Obviously, the funds will be used to push that into the clinic and beyond. In addition, behind that, we have another rare pediatric liver disease using this time a different type of editing. It's called RT editing. So we take a nuclease and couple it to a reverse transcriptase. And what that allows us to do is effectively rewrite sections of DNA. So if you have a specific mutation you need to fix, you can go in with this technology and rewrite it. And so again, focused on a rare liver indication where we can go in and fix the underlying mutation and hopefully provide significant benefits to these patients. So that's the two programs in the liver. And then we have a couple programs in the CNS. One of the benefits of our technology is it's very small, so it allows us to move out of the liver. Currently, the rest of the CRISPR companies are really in the liver because that's where they're, kind of have to be because their technology is large. So we can move out of the liver, and we really are intrigued by the central nervous system. There's a variety of genetic diseases there that have no effective treatment. So we are starting with a couple of programs targeting ALS, which is a very deadly, terrible disease for which there's effectively no real treatments today. And so being able to hopefully impact those patients positively would be fantastic. So from a proceeds perspective, it's really about bringing 3 of the programs into the clinic. So PH1 being the lead program, and then 2 additional programs into the clinic is going to be the primary focus of the fundraise we did.
So you serve as a board member and former chairman of ARM, which you were speaking about before. Can you talk us through your role there and how some of those ongoing conversations that happen with the FDA as well?
Yeah, so I think as chairman of ARM, it's been interesting to be involved with the organization. So ARM is made up of about, I think, 415 or so companies in cell therapy, gene therapy, and gene editing. And the primary goal is to really help advance the industry and help in areas where there's any sort of bottlenecks or concerns. And so the 3 big areas I think where ARM is able to impact and help the industry, one is in the regulatory side. So by identifying areas of interest that where we can help the regulatory agencies educate them or partner on initiatives, that's really helpful. Is one area. Another area is in really lobbying. So they do a fly-in every year into DC where there is, we'll go in with specific asks across the members of Congress. Last year, there were a couple of asks. One is focused on creating better reimbursement approaches for these one-and-done approaches, because one-and-done treatments historically, didn't exist, and suddenly we can cure patients. The question is, how do you then get reimbursed? Because the system's set up for reimbursing, every monthly you go get your prescription and it gets paid for. In this case, it's once and you're done. So how do we reimburse that and put in the right sort of framework to do that? So there was some work on that was done with the CMMS particularly. And then importantly, ARM is a global organization. So similar to the work being done in the US, it's similar work done in Europe and Asia on trying to make sure that we help the regulators understand the unique opportunities and challenges with these types of therapeutics, and making sure that we put in place the appropriate frameworks to ensure patient safety, but also make sure we eliminate hurdles on the regulatory side that may not be useful or are not relevant to these types of therapies. So that's, I think, the primary, some of the big missions of ARM. They also have one of the, I think, the premier meeting within our industry every year, and it's called the Meeting on the Mesa, where I think about 2,000 people typically go, and it's a great opportunity to sort of understand what's going on in the sector and how these therapies are really changing patients' lives today and will continue to do so in the future.
Very good. And just a question back to you then. So you've been on both sides of the fence, big pharma, small biotech. You're at Pfizer for almost 8 years. Talk us through the difference between being a leader in a big pharma versus a small biotech, in terms of how they think, just the main differences.
So that is a good question. So I think big pharma, you're looking at companies that are, they're large. They have a lot of different governances. There's a lot of committees, and everything moves a little slower. And so you have to be able to navigate that system, and being able to move quick is very challenging, I think. And so, when I moved over to biotech, I actually found it to be a kind of a breath of fresh air to be able to move so quickly, to make decisions and move fast. The downside, I think, to being in biotech is you don't have these large organizations to do things. So when I first joined Sigilon, as I think about the 10th employee, we're like, okay, we don't have regulatory. I guess I'll own that. We don't have quality. Well, okay, I'll own that. So you have to wear a lot of different hats, which if one is, if that's part of your DNA, you love that, then you will thrive in biotech. If you're used to having, this huge organization you rely on for all the expertise, then that's not a good fit for someone trying to transition from a big pharma. So very different. I think the other piece that I find interesting is in big pharma, you never know when there's going to be a reorg or a strategic refresh that's going to impact your group or the work you're doing. And so you're kind of, you just work along, and someday someone somewhere decides, makes a decision, your group disappears. I think in biotech, you often have a better window because particularly in, smaller companies, when the money runs out, what you have to accomplish, and it helps you to focus very much on what needs to be done. And if if the science doesn't break or you don't accomplish what you're supposed to, then obviously then your role may be impacted. But you have I think, you control your destiny more so, I think, in a small company.
Yeah, I think that's a really good way of putting it. And I know one of the areas you wanted to talk about was about a Chinese scientist, Zhang Qiu, who was jailed for 3 years after he'd said he created the world's first gene-edited babies. Give us your perspective on how to think about the ethics of gene editing and whether we should gene edit embryos.
Yeah, so if you think about, gene editing is a powerful, powerful tool. It allows us to really fix problems or mutations in the DNA. It has only been used in probably 200 patients, maybe 300 to date in vivo. And so the way it's done today, for example, our therapy will edit just the liver cells, which is where the disease is. And that is appropriate. And I think particularly in these rare diseases that are extremely deadly and have terrible outcomes, that's where we should be focusing today. Someday, CRISPR use will be more widespread in other diseases. And eventually, I think it will be utilized a lot like a vaccine, where you'll have your DNA sequence. They'll notice you've got a propensity for Huntington's or Alzheimer's or whatever it may be, and you'll get it fixed before you have the disease. So that's all called somatic cell editing. So it's you're just editing cells of the body, not germline. Germline editing is when you go in and you change an embryo or the DNA in a sperm or egg, fundamentally changing the DNA for all subsequent generations. Now, the reason this is problematic, and it's called heritable genome editing, is that a couple things. One, we are just learning how to make these things safe. I think they are safe for use in specific organs today. I think we don't know and can't prove safety in editing an embryo because we don't, you'd have to do like 3 generations or 4 generations in animals to confirm it's safe, which is a 20-year study. That's not really feasible. And so I think particularly today, if you think about patients who have a disease and they don't want to pass it on, there are approaches today to allow you to not do that are not involved germline editing. And so I think the view that I have, and I think most folks in this space have, is that there is no need for germline editing, that we can accomplish what we need to in the somatic cell editing space, and that's where we should focus our efforts. I think particularly as you get to the ethics of it, I think the biggest concern is if you start editing the embryo, is where do you stop, right? You start having designer babies. So, and so that's really, I don't think, the purpose of the technology, and nor is that something that we should strive to do. And so I think, particularly given all the unknowns on the science side and the technology side, I think we are best placed to not do any heritable genome editing in the near future. It's unfortunate that this one scientist in China did this, and he did this on 2 patients or 2 babies. We don't know the outcomes because Chinese government's been very quiet about it, but it is I think ethically wrong, and I think it's too early to begin doing this type of work. So fundamentally, I think we as an industry need to focus on making drugs for patients with severe diseases where we can impact this particular disease in that patient, but not targeting any of the germline.
Awesome. And then this is a question that we always finish with on the BioHub. If you were to have dinner with 3 people, past or present, who would they be and why? Don't necessarily need to be biotech innovators, although they can be if you like.
It's 3 people. It's a good question. I've thought about this, and I think the 3 I would do would actually be historical figures. One would be Isaac Newton, who I think is fascinating. When I lived in Cambridge, the first Cambridge, we'd often go to Westminster Abbey down in London, where he's buried. And, I think he was a Cambridge faculty member, I believe. And it's fascinating to see his life and the work he did on the science side and the amazing discoveries he did with effectively very rudimentary techniques and instruments at his control. The other one is Da Vinci, who I think is fascinating, right? Here's a guy who has done some of the most famous paintings in the world, yet was probably one of the most prolific inventors that could see the future, right? He thought about flight. He thought about these things hundreds of years before it's feasible. And so it'd be fascinating to sit down with him. And then the third one is Abraham Lincoln, who I think is a really good lesson in leadership in hard times. And I think there's a lot of good learnings, I think, that, he's had, and his experience would be fascinating. So those would be my 3: Abraham Lincoln, Leonardo da Vinci, and Isaac Newton, because I think very different perspectives, but fascinating characters.
Yeah, but look, 3 terrific answers there. And thanks so much for coming on and helping us out. I think it's been really interesting hearing your perspective on, Arbor, the gene editing market. So thanks so much for coming on. It's been a pleasure.
Thank you, Tom. I appreciate it. And it's a pleasure to have the conversation.
Awesome.